Okay, our final contestant today is Marie Adomako, who's also a biological sciences PhD student. And so please join me in welcoming Marie. Oh, her advisor is Professor Susan Golden. So please join me in welcoming Marie.
These are cyanobacteria, affectionately cyanos. 2.6 billion years ago, these tiny bacteria invented photosynthesis, pumping the atmosphere full of oxygen and changing the course of life on Earth. They're still at it today. About 30% of the oxygen in the atmosphere comes from cyanos. They're good candidates to modify to be able to produce biofuels and food supplements because they're very efficient. They eat sunlight, and they're very simple. The entire organism is just one cell. They're grown in large outdoor ponds where they can literally turn sunlight into food and fuel. Now, cyanos can live a couple of ways, either suspended in the water or in a biofilm. A biofilm is a community of microbes that attaches to a surface and starts producing a sticky matrix to protect itself, and that makes it very difficult to remove, which we're all very familiar with because we have to brush biofilms off our teeth every day in the form of plaque. We don't know what makes cyanos choose to live in a biofilm rather than by themselves, and so that's what my research asks. How do cyanos form biofilms? Because it has practical applications. If you can find a biofilm off switch, you can prevent buildup in water treatment systems or other aquatic environments. Or if you can find a biofilm on switch, you can grow useful cyanos more efficiently and using less water than the usual methods. So to find these biofilm on or off switches, I have to look at the genes of the cyano. In my research, I have two different cyano strains. They are the same species, and they are 98% identical genetically, and yet one of them keeps the biofilm switch off. It doesn't stick to the flask when you grow it in the lab, and the other keeps the biofilm switch on. By comparing them, I can narrow down my hunt for the biofilm on or off switch genes to the 2% difference between these strains. But I still have to find the exact genes. And to do that, I'm using a very classic technique. Break it, see what happens. In each of these strains, I have a collection where each cyano cell has a different gene broken, as well as a unique DNA barcode, so I can easily track it later with sequencing. When I grow the collections, I can track which gene, if it's broken, will turn an off switch to an on switch, or vice versa. That lets me connect the gene with its function in biofilm formation, and maybe use that gene product to flip the biofilm switch. My approach looks at the question from two opposite directions in order to see the whole picture. And eventually, I'll be able to turn cyanobiofilms on or off with the flip of a genetic switch.